Okay, I seem to be online. Good morning, everyone. I see some 34 people online. Shall we wait for a minute more? It's already 11:03 as per my system. Y'all should be punctual. Yeah, but I suppose you need a 5-minute break between teachers. So we will move on to the next system, right? We're talking about systemic anatomy. Yesterday we did joints or arthrology, the study of joints as a system and as individual types with examples. I hope you've had the opportunity to revise some of that at least, and that you're in the process of getting your books. I've asked your Vice Principal, Dr. Negi, to publish the list of anatomy books that we recommend, either to your student group or to your mailing list. I don't know in what format we communicate with you as a group. I hope you have access. Sooner or later, all of you will have to elect a class representative, and one or two ladies and gents or whatever. And it'll be good if that person can represent your requirements, any issues, any concerns you have, to the faculty as a group, and then we can address it efficiently. Okay, so now I have 91 people online, and let's start. Today we'll talk about the this is what we finished so far. I haven't taken cartilages as a separate class, but I did mention that it is a very unique, dynamic, supple, yet protective, versatile tissue which we have divided into three types. That is hyaline or glass-like cartilage, which is almost transparent, elastic cartilage, and fibrocartilage. Fibrocartilage examples we saw in yesterday's class, such as the intervertebral disc. But these four things together, bones, cartilage, joints and muscles, are together forming the system which we know as a musculoskeletal system, and you all have had a sequence of classes describing each of these. Today we'll go to the next one. It is also a set. The circulatory system, I think your physiology lessons have already talked about blood and circulation as a physiological phenomenon which keeps everything alive and keeps everything going. And it is underlaid by principles which you have studied in general physics perhaps. But those principles of fluid dynamics applies to the fluid tissue we know as blood, which is everywhere and does a great many things and needs to be kept in a movement, following or obeying physical laws such as gradients of diffusion, but also having unique mechanisms which allow its flow, that is the flow of blood as a tissue, into and out of what we know as intravascular and extravascular compartments. So that is physiology, but to make that happen we need a structure. We need an underlying architecture of materials which will lend themselves to those functions in a very easy way. That means the material characteristics will have to be such that the functions of blood flow will be served, right? So our circulatory system has evolved beautifully to serve that. First, we have a network of tubes and pipes in which this fluid will flow. Then we have two halves of it, because one half will flow with what we call oxygenated, nourished blood, and the other half will flow with deoxygenated and denourished, if you like, or blood which is full of waste materials. It's the same blood, but certain components of it have been extracted out by each and every cell in your body, and cellular effluent, sewage, waste has been thrown out, which includes carbon dioxide and every outcome product of metabolism which the body cannot use. It's thrown back into the same liquid, but therefore cannot enter the same tube it will enter a different tube and go to a different place so that it can be dealt with, then replenished, refreshed, and again sent back into circulation. It's sophisticated par excellence. To bring it about, these tubes have to be supple, they have to be strong, they have to be able to take hard pressure, they have to be able to relax into almost negative pressure to allow these things to flow out. So the structure of this circulatory system set of tubes has to be so exquisite. This is all just to bring about the adult circulation. But from a fetus, when you're developing stage, the demands of circulation are very, very different. So fetal circulation is a different ball game, but you can't be replacing tubes and reconnecting things in a living body. So an architecture, a system has been laid down, which we, at the moment of birth, can change within seconds and then over a few days to reset the direction of the flows to allow adult circulation to happen, where oxygenation is coming not from the placenta but from a newly independent, extremely vital organ called the lungs. So the heart in the middle and the lungs in the middle are going to work upon or are going to contribute to this circulation in their own unique ways. The heart essentially working as a muscular pump, and the lungs being the whole purpose of it. The lungs taking in oxygen being the whole reason why the circulatory system was set into the place to deliver that atmospheric oxygen into each and every cell for the metabolism that it has to do. With so much introduction, I will just outline what forms these tubes and what we call them, because all the terminology is probably new, and what we understand of the mechanisms and the separate characteristics of each kind of tube. We'll look at that further in histology. When we do microscopic anatomy, we'll actually look at that and then we will revise it. But it will be very important for you to remember this, the basic plan, the overview, the grand picture, when you dive into dissection also, because as soon as you open the skin in a live person, first thing they do is bleed, which means you've disturbed their circulatory system at a superficial stratum and allowed that liquid tissue to escape its boundaries. Right? So we must know how much is permissible, how much is useful to us, how much can we put in the service of repairing this patient of ours, and how much must we protect and take care of and re-establish back to its anatomical structure so that the patient can heal and go home. So for the background of that, all this is necessary today. Your cardiovascular system conveys nutrients, oxygen, hormones, etc. to everyone and everything, all living tissues in your body. And it redistributes and disperses heat. It's an extremely important thermoregulatory component. The organ which is bringing about heat dissipation is the skin, but the blood supply to the skin is what is going to allow this dispersal of heat. You sweat, you become hot, right? So that is served by bringing blood there, allowing its heat to dissipate, therefore cooling it and then returning it back to the core. The heart functions as one, it looks like one organ, but it is two separate pumps. One pumping into the pulmonary circulation, heart to lung to heart, and another pumping into the systemic circulation, heart to everybody else, from brain to your tiptoes. Arteries are named as those channels which take away blood from the heart, and veins are those which return blood to the heart, not necessarily oxygenated or deoxygenated. I've colored them red and purple here because as a majority, if not otherwise specified, arteries will carry oxygenated blood and veins will carry deoxygenated blood, and that's how you will color them in your record books also. There is a color code we follow for medical illustration everywhere in the world, and that has to be followed by you too. So whenever you're drawing an artery, you'll color it red. Wherever you're drawing a vein, you'll color it blue, even if it's not carrying that kind of blood in it. That happens particularly in the fetal circulation that I mentioned, that the same tube for some part of its life is carrying the opposite kind of blood, but that does not matter because it serves its correct function at the correct stage of life. We will come to that in detail when we do the fetal circulation. Every tube that we have has this particular structure. The larger blood vessels have these structural features. A large dominant component of it is muscle. You remember we said there were 3 types of muscle. We said there was skeletal, which we dealt with in detail for the musculoskeletal system. Then we said there was smooth muscle or non-striated muscle or involuntary muscle, smooth muscle. That is those spindle-shaped cells with the central nuclei. So a whole bunch of smooth muscle tissue is provided in the walls of our blood vessels, especially the larger ones. And that area where this is supposed to be smooth muscle, the light pink zone, right? So a good, if you take the diameter of the tube from here to here, the thickness, sorry, then most of it seems to be pink, right? So a good amount of the thickness of the tube is muscles. In other words, you can say arteries are muscular tubes. You wouldn't be wrong because dominantly that's what they are. But we further named and classified the wall of every artery into 3 zones. The most inside one, the blood is flowing in this empty space, right? So the most inside one is lined by a very, very, very smooth layer of cells. Now I haven't done histology with you, so talking about these particular structures at this point, I don't know how many of you will be able to grasp, but I will tell you the terminology anyway. Later we will repeat it and explain it again when we do histology. Don't worry about it. Try to get familiar with the terms. So this innermost stratum, which is extremely smooth on the inside, is called the tunica intima. Tunic means coat, right? Intima is insidemost or most intimate, most closely associated with the flowing blood. So this tunica intima, can you see, is the purplish grey area. The innermost lining of it is living cells in a single layer, right? An endothelial layer, the endothelium of the tunica intima. Lumen means the empty space. It's not empty, it's always filled with the blood tissue, but here we're showing it as empty because we're focusing only on the tube. Then we have, after the blue-gray zone, which is composed of this inner endothelium and some purplish material, light purple, dark purple, little bit of material. These together is called tunica intima. Outside of which there is a sheet of elastic material, the yellow, which is called the internal elastic lamina. Then a whole bunch of pink material, which is the tunica media. Media means middle layer. So tunica intima, purplish, tunica media, light pink, and tunica adventitia or externa. In this figure it is green. It is very fibrous. It is almost entirely filled with fibers laid down in strap-like groups, right? Now this fiber, though we even to a muscle we said muscle fiber, so that is very different. A muscle cell is a living thing which can adapt and which can do contractile function. Here we are talking about fibers which are neutral extruded long proteins, collagen, elastin and reticulin. These are long chain molecules with interlinks with each other, which are created by a cell called a fibroblast and then thrown out of the cytoplasm. So wherever they're thrown out, they're laid down in that particular diet. They can't do anything, they're not alive. So this whole bunch of fibers are going to be continuously placed in the direction, in the thickness, in the dimension required by a bunch of connective tissue cells called fibroblasts. We are not concentrating on that today. We are only saying that the tunica adventitia of this blood vessel is the outermost layer and is always fibrous in nature. So this tunica adventitia is the outermost layer. This plan, tunica intima, tunica media, tunica adventitia, is the general plan of any vessel, anywhere, at any stage of its life. That's how it's going to develop. So the tiniest vessels of the smallest dimensions, we call them capillaries. I think you already know that. They won't have a media externa, but they'll have the tunica intima structure. They'll have an innermost endothelial lining. So a capillary, 4-5 capillaries are coming as branches of one arteriole. That arteriole will develop a media and a little bit of an adventitia. 5-6 arterioles together are coming from one big artery. That will have all the 3 layers very well laid out like in this diagram. So the essential structure of the tube is the same. Depending on its dimension and caliber, the emphasis of the external layers increases. Right? So we will look at that in the next slide. But in this slide, what we are saying is any vessel basic structure is a tunica intima, a tunica media, and a tunica adventitia. This media is separated from the intima by a internal elastic lamina and from the externa by an external elastic lamina, the yellow, mustard yellow zones. So it's like looks like cheese also, right? So the inner elastic and the external elastic sheet separates this bunch of muscle, this slab of muscle, from the adventitia outside and the intima inside. Okay, and in the outside layer of this fibrous bundles, in between will be blood vessels, nerves, lymphatics, all kinds of things which are included, because the blood flowing inside the lumen cannot perfuse its constituent nutrients and oxygen all the way through the thickness of this vessel. It's too much thick for it to simply be able to perfuse, right? The innermost little bit of a few millimeters can be kept alive by the blood within the lumen, but for the rest of it you need blood of its own. So it is a vessel to a vessel. It's called a vasa vasorum. Vasa vasorum is a vessel, an arteriole, a very tiny arteriole branch and a very tiny venule tributary, which is serving this thick vessel itself. You'll see that. So these are vasa vasorum serving this item, which is a muscular artery. Then you have lymphatic vessels. Sometimes here in this diagram they're colored purple, but usually in life, in dissection, in microscopy, you don't see them at all. Lymph vessels don't take up any stain. They are even finer dimension and even finer tissue, thick wall than the blood vessels. You hardly get to see them at all, but they do exist, and lymph is a very important phenomenon. We'll come to that. The endothelium, we said, which was the innermost layer, which was part of the tunica intima, is lying on this light purple continuous domain, which is the which is called the basal lamina, which means that the cells, the living cells called endothelial cells, are lying on a layer which is called a basal lamina, which is a non-cellular layer. It's an extruded matrix material, just a sheet, very thin sheet. In this case, it is, you can see, light purplish sheet. And outside of that, they gave a subendothelial connective tissue purple domain, which means a layer of fibers. This purple is also finer, much finer fibers, not so thick as the external fibers, which is separating this endothelial stratum from the next stratum, that is a tunica media stratum, on the inside of which is your internal elastic lamina. So it is a kind of layer, sheet by sheet by sheet by sheet, each of which is having a reason for being there. We'll come to that in histology. The types of blood vessels now will depend on their function. I think already your physiology class has touched on it. The resistance vessels, they are going to resist. Their function is not to resist, but their very structure, the fact that they are so tough, makes them resistance vessels, and these are the arteries, and particularly the arterioles, and they are the ones which power is measured by a blood pressure reading. The exchange vessels are very, very important because they have to allow exchange of oxygen, carbon dioxide, and glucose and waste products. These are the fine caliber but huge network vessels. If you see any one capillary, it's very fine. It's like a hair, thinner, much, much thinner. But if you put all the capillaries together and open them out and lay them, their surface area is huge. That surface area allows intimate contact between the molecules which are there in the plasma of your blood and which are there in the intracellular and extracellular compartments, right? So exchange is possible. They're called exchange vessels. Capillaries, which is one type. Sinusoids, which is a wider bore, that means the lumen is larger but the lining is the same, and small venules, all of them behave as exchange vessels. And capacitance vessels are those which have a low pressure, can take large volumes, they can expand and fill up with large volumes of blood, and these are your veins. Arteries, veins were decided on whether they were going away from the heart or whether they were coming towards the heart. Arteries will be named as conducting arteries, which are elastic in nature, which have the capacity to stretch a bit to allow blood to flow into them. So they will conduct incoming blood in some direction, and they will be large in diameter. Distributing arteries are of a more muscular nature, as we saw in the previous diagram, and they are of a medium diameter. And the resistance arteries or the resistance vessels are the arterioles, which are branches of the arteries, which are going to not do very efficient conduction, not do any distribution at all. We are calling them resistance because that's how we see them, but they are basically distributing to some extent the whatever the artery is bringing into it. They're just sharing and increasing the territory of any one artery by becoming branches of it. So this is our understanding of how arteries can be viewed. Veins are much more thin walled. Their adventitia is dominant. The media is very minimum. There's hardly any muscle in the wall, though there is a layer called a tunica media, and they have a lot more elastic tissue rather than muscular tissue. They also, the large veins, and they have, because they're such low pressure zones, they may not be able to resist a flow from the opposite direction, backflow. So they are provided with a mechanism, a flap-like mechanism, where that endothelial lining in the middle is folded into these flap-like extensions inside in a semi-cup kind of fashion. So they allow blood flow in one direction, but if the blood falls down for some reason, this cup-like thing fills up and closes the gap, the area within the vein, not allowing to go back into the distal segments. Important provision, mechanical provision. There's nothing contracting and holding it, and not that kind of a valve. It is just a flap of tissue. Actually quite brilliant, that just by providing an extra flap of tissue, the venous system manages to not have backflows of blood. So veins have valves, especially in the limbs, especially in the lower limbs, because in the erect posture, which is natural waking posture of a human being, blood will, by simple physics, will tend to pool by gravity in the lower limbs. So it must be provided valves, so that whatever is coming back, the volumes are building up and flowing back towards the heart, but against gravity. Whatever manages to reach up will not fall back down again, but will keep getting brought by various mechanisms, osmotic pull, muscular action around the vessels and so on, back towards the heart. Capillaries are the smallest caliber vessels. They may be continuous, where their endothelium is uninterrupted. It is complete, 360 degrees, it is a complete layer. Or it may have apertures in that endothelial layer, when it's called a fenestrated capillary. If the dimension of the capillary is much bigger than 7 microns, then it is called a sinusoid. So capillary will be 7 to 8 microns. Your RBC caliber is 7, isn't it? So actually RBC squeeze through capillaries one behind the other, like soldiers marching in a row, which is a good thing. That is a clever phenomenon, because then RBC is forced to be exposed on all its surfaces to the thin-walled endothelium-lined capillary, and exchange can take place in all directions. Plus it slows down the flow of blood, so that one by one the cells move through it, and each and every of them can exchange their oxygen carbon dioxide as they pass through. If it's fenestrated, other things apart from extremely small molecules like oxygen and glucose, other things can also go off through the holes, the gaps between the cells which are forming the endothelial lining. That is the purpose of fenestrated capillaries. They don't exist everywhere. In the special places where they are there, we will tell you as and when we come across. Sinusoids will have a much wider, 2 to 3 times the luminal dimension. So the 8 to can go up to 20 microns, so that more number of cells can flow through, and this wider space allows for more blood to be able to flow through this. And it is also a very sluggish zone. There's no fast flow through sinusoids, but it allows a good amount of blood to be held in that organ for some period of time, and particularly allows big molecules to come in and to work in that particular tissue. We will show you where and when sinusoids are available in the body. So this is just a repeat of the same picture without the labeling. I could do an exercise of this, but I want to close a little early today. But find this in one of your books or one of your any website, wherever you go, and try to remember the names of the zones as I have taught you. Some concepts and definitions broadly in this overview of what we are calling the circulatory system. You have a name called venae comitantes. Venae. Comitantes are those are going along with, together, right? So sometimes an arteriole will be one in nature, but the veins accompanying it will not be a single vein, but two veins, one on either side of it. This happens in many areas. So the arteriole or the arterial vessel is of a larger caliber, and the blood is flowing in one direction, away from the heart. And the returning blood, which is deoxygenated and all its nourishment has been taken away and waste material has been added to it, it could have returned via one vein, and then we would call it the vein of the same name, right? For example, femoral artery, femoral vein, right? But in many areas, instead of that, there is one singular artery, but 2 or 3 venous vessels accompanying it, bringing back the blood from the same territory of the artery. Then it's called vena comitantis, and it usually forms a neurovascular bundle. There will be artery, there will be 2-3 veins, usually 2. And there will also be the nerve of that region, which is going to serve the motor function for musculoskeletal system, the sensory function from the skin, from the joints, from all the surrounding areas, as well as the autonomic functions. Its branches will also be keeping up the tone of these vessels. It will also be doing any other autonomic functions, serving that limb region or that organ region or the body cavity or wherever it is. So we will mention this to you. You just remember that there is a term called venae comitantis. Then you have the counterflow heat exchange system. This I think physiology must have taken in detail, so I won't again repeat that. But the fact that we have vessels traveling with other vessels and other linear structures doesn't allow sharing of contents of the tube, but allows heat as an energy flow between them. And this is an important phenomenon utilized by the body in thermoregulation. Counterflow ion exchange mechanism. Ions are really tiny. They don't even require much of a mechanisms to exchange between systems. But our cell membranes and our arterial walls and our tubular structure, whatever they may be, do to a large extent separate the domains, intravascular, extravascular, and so on and so forth. But ionic exchange can take place when things are packed close together, across boundaries. Sometimes ionic movement can take place, and within juxtaposed, very close together, intimately situated compartments, right, physiologically. Microvascular bed, where you can imagine or you can conceptualize that a region of tissue. Now we are not thinking systemic, not system-wise, but a region-wise. This bunch of tissue, this bunch of tissue. Inside of that, the roads, the arterial roads, are already laid out in a certain way, right? And they serve those surrounding tissues. This much of muscle, this much of bone, this much of skin, this much of whatever else is available there, and their branching pattern will therefore divide, and that network will share that whole territory from that parent vessel. Similarly, the waste, etc., will come back to a particular vessel. Similarly, there will be a nerve supply territory. There will be a lymphatic drainage territory. So the if you look at it from the cellular point of view, that whole bunch of different kinds of cells which are available there in that tissue or an organ, which are perfused by a singular artery vein system, can be thought of as a microvascular bed. The network is a bed in between which different tissues are set, and this thing takes care of all of them together. Right? That's one way of actually that's a useful concept to have in your head, particularly in your organ systems. Like, let's say there is a microvascular bed supporting the pancreas as an organ with all the things that it does, right? Or there is a microvascular bed which is taking care of the chunk of your thigh muscle, and so on and so forth. We will come to that, but think of, now you're not thinking of separate tubes forming separate branches, but you're thinking of what word can I put, like a scaffolding, like a service of vasculature, and the service provision is by our vascular network, including the finest capillaries, and service is being received by all the other tissues which are non-vascular in nature, something like that. Okay, then there is a concept called portal system, which might have been mentioned again in yesterday's physiology class. The venous system particularly, sometimes a vein comes, usually an artery comes, breaks up into arterioles, that breaks up into capillaries, then they reform and form venules, which forms a big vein. If that vein goes and breaks up again into a network and again forms a vein, right, then it's called a portal system. Portal means actually door. So it's like it is venous blood, because it is finally going to converge and go towards the heart, but it has a two breakup system rather than a singular breakup into capillaries, and we will see where it is provided and why as we go along. Then sometimes the opposite also happens, especially in the kidney. We will see that there will be a capillary network. We expect that the incoming blood is oxygenated and the outgoing blood is deoxygenated. That's the whole purpose. But sometimes the afferent incoming blood is arterial, the outgoing blood is also arterial, which means an artery has broken up into a capillary system, and the efferent from this capillary system is also entering an arteriolar system. Both are oxygenated. So something else has happened over there, looks like, right? Not like the typical arterial capillary venule system. So this happens sometimes in certain areas, we will see. Then arterial anastomosis. When an artery breaks up into its many branches, it... Are you in Srihari's meeting? Yes. So this anastomosis will, this arterial network must become capillaries and then become veins. But sometimes the artery, the big artery itself, it may be undergoing a capillary anastomosis, but on its own wall somewhere, it may be meeting up with another artery coming from somewhere else, and the ends of the two arteries may fuse with each other and become one continuous arterial tube. That's called an end-to-end anastomosis. While the branches of this one and the branches of that one are doing their own thing. They are spread out into the tissue and supplying their own territories. But the artery, instead of ending in a tapered capillary system, ends by fusing with another one, right? And that is usually of a convergence kind of thing, where it becomes smaller and smaller in caliber as its branches are given away. And somewhere near the smallest size, there will be an end-to-end anastomosis. Sometimes there will be a convergence kind of anastomosis, where the smaller branches of two territories get shared by the arterial branches. Sometimes there will be a transverse anastomosis, which means side to side. We will look at each one when we do gross anatomy. When you do the dissections, you'll actually see these anastomosis. This is important because we utilize this in surgery. If an area is getting branches from two different arteries, we can tie one. We can sacrifice one side supply because the other one is there. right? Or if there is a difficult situation because we're going to take out a chunk of tissue in surgery, we're going to excise it, then whatever is remaining, we have to leave its arterial supply behind, otherwise it will die. So then we may have to create an anastomosis, right? Manufacture. We have to style an anastomosis, which is non-anatomical, but we have to leave it behind so that whatever we've left behind for the patient is completely vital. It is perfused and it will live and it will work. This brings us to the concept of an area which is the territory of a particular artery, can therefore be called an angiosome. Just remember the word for the time being, where we are saying that this is your territory, right? Nobody told us that. We did these studies and figured out, okay, this particular artery bringing blood from this particular place is going to take care of this much chunk of tissue or this much area, which includes this organ, this part, this bone, this muscle, this joint, whatever it is. All of that together would be an angiosome because it is a vascular territory. And another important concept called collateral circulation. This means that any one territory may be having a one main artery and maybe one or two other 3-4 minor supplies also over and above. And in normal life, in normal function, when it is healthy, all of them are perfusing the blood. The blood is the same, so it is getting perfused generally. But if you are going to, for some reason, some disease process, some pressure has happened, some tumor is growing, one of those arteries is getting slowly compressed and its blood flow is stopping or stops. It can happen over a period of time or it can happen suddenly. Then, if it is slow especially, the remaining ones which are supplying the territory will grow new branches or open up the diameter of their existing branches and feed the tissue, keeping it alive. Which is great, it is nature's way of correcting itself, but on the other hand, the patient will not get any symptoms because that tissue is still active, is still vital. And we will not be able to diagnose, we won't even be able to suspect there's anything wrong because collateral circulation is taking care of the keeping it alive, the perfusion of that tissue. Some areas, no collateral circulation has been provided. That whole cone of tissue has only one artery, and if that's going to be compromised, that will die. That's what gives rise to what we have known as infarction, where one artery is compromised for whatever reason, external pressure, internal thrombus, anything, and the entire amount of tissue which is supposed to be perfused by it suddenly is starving for nourishment, and that is therefore prone to just die. That's called an infarction. So you can have a myocardial infarction or a cerebral infarction. We will come to that later. You remember collateral circulation is an important phenomenon. It's provided in many areas. In some vital organs, it's not provided at all. We need to know which is which, and we need to know what to do if any disease happens. A microcirculatory unit is again a concept where we are saying that between the terminal arteriole and its branches, which is essentially a capillary, which will lead back into a venule, which will lead back into a connecting venule, there will be certain channels where there is free flow. There is no constriction and flow is continuous and not interrupted anywhere. But in other places, or normally, before this arterial branch becomes a capillary, there will be a pre-capillary sphincter, and then at the venous end there may be, it's not named here, in a few places there may be a post-capillary sphincter. That's how we've named it. It's not there in this picture. The point being that through certain mechanisms, at sometimes you don't want this much distribution of blood. For example, the blood volume is falling for some reason. The circulatory system will decide not to fill up its capillary network and lose the blood or lose the heat or whatever it is, and all precapillary sphincters will get operated and they'll get closed so they don't fill up the capillary networks. But even at that time, these thoroughfare channels, they don't have that sphincter mechanism, so they will have continuous flow. So some amount of perfusion is still kept alive, but the larger amount of perfusion is held back. for various reasons. So this is all under autonomic control. We won't go into the details, but remember that there are phenomena called meta-arterioles. There are terminal arterioles which have branches which are mainly capillaries protected by precapillary sphincters, but also some thoroughfare channels which do not have a sphincteric mechanism. Arteriovenous anastomosis is another very important phenomenon where there is no capillary in between an artery and a vein. So you normally have artery, then you have arteriole, then finer channels, then they become capillary, then there'll be a network. Slowly the red will become purple, and then they will come back into the vein. This is a normal plan, right? Lots of distribution and lots of coming back. But in certain areas, a branch, if you follow one of the arterial branches, there is no capillary, and some exchange, some through simple diffusion mechanisms may happen through the wall, and then it will come back and join the vein. These are important. They are found in nail beds, in the digital pads with fingers, and they form what is called a glomus organ. The plural of glomus is glomera. The point is that these AV anastomosis exist for us in certain places, and the function is that you can shunt off the blood from arterial to venous without allowing it to fill up the peripheral channels. Very importantly, utilized as a heat saving mechanism, thermoregulation, because they are all present in the integumentary system areas. Nail beds, digital pads are in the skin. Because your skin is such a huge surface area organ, it functions very usefully for maintaining your core body temperature. So if it's going to be very cold and you're going to allow your skin to perfuse normally, then you're going to be losing more heat from the surface, which is why you'll withdraw that by operating the AV anastomosis. You'll open them up. Under normal circumstances, they may not be open so much. They're always potentially openable. And when you open them up, the blood will not go into the capillary networks, which is why the skin becomes pale. It becomes white. When it is cold, there's a blanching. Blanching means a lack or withdrawal of circulation to the surface skin, which you can see, especially in thin-skinned, fair-skinned individuals. You can see that the pinkness of the skin is reduced to a whiteness or a paleness, especially as a cold protective mechanism. And the way it operates, how it is brought about, is by operating your arteriovenous anastomosis. Apart from that, there are other functions. We will look at that later. This is just to show you. Remember, I showed you an intima where we said there's an innermost layer of cells, and we saw, spindle-shaped cells with round nuclei. That is how you see it in cut section, and that was a painting. This is an actual photograph of the internal view of an artery to show you that the endothelium, if you look at it, if you open an arterial tube and look at the inside layer, this is how the cells are laid. The cells are elongated and so smooth and so tightly juxtaposed. Cell membrane edges are flush with each other, forming an extremely smooth surface. So there is no impedance to the flow of blood. There's no friction. There's no possibility of friction. So this is a scanning electron micrograph. The tightly packed endothelial cells are elongated in the direction of blood flow. The shape of the cell also adapts itself, because I wanted to show you this. Firstly, it's a picture of a living thing, and secondly, there are no colors when we do that with our much higher magnification micrography. And thirdly, that whatever tissue you see has a etchy stained glass slides, which you can put in a shelf for 50 years. Actually, in life, in your patient, in you, they are dynamic systems. They are already adapting to what it is that they are supposed to do and what is happening to them on a daily basis. So this endothelium is in touch with flowing blood all the time. It is adapting to the pressures and the volumes and the stretches of that blood, to the requirement, the oxygen demand of the tissue which is to be perfused, to the condition of the heart which is pumping. All these dynamics are operating upon it and keeping it alive and responsive. The other thing I wanted to show you is that we see, or we draw, capillary tubes as very uniform, very very pretty tubes of an even caliber, but the capillary wall is like this. Can you see the cells forming it, the endothelial cells forming it are looking a little rough. They're all living cells. They will have a little bit of a fibrous network. They'll have a cytoplasmic membrane. Over and above that, they will have these extra cells called pericytes. Here, pericytes, which support the vessel wall, and they have these foot-like processes. All these pericytes, which are supporting this capillary, this tubular capillary wall. So, just wanted to give you a little live look at what your finest blood vessels look like and operate like. What is a pericyte? What does it do? Why is it there? We'll look at it in histology. Okay, so I've come to the end of my class. I'll close here, and tomorrow we'll look at another system. I'm leaving you a little early, consciously, so that you get a break, and also so that you will make the effort to please access reference books or textbooks or whatever it is, and take the time to either rest, refresh, or study by yourself. By now you must be already setting a schedule for, okay, 10 to 12 is my class, rest of the day what am I going to do? You can just celebrate the fact that you're a medico now, that you managed to get your seat. Do that by all means, but also schedule some time for self-directed study. When I was an MBBS student long time back, 40 years back, more than that, 40 years back, I had a classmate, and he calculated that apart from what we are doing as college work, right, we had classes from 9 to 4. Every subject, anatomy, physiology, biochemistry, requires 2 hours of study time per day. How he came to that conclusion, I don't know. We thought, oh my God, like, will I ever have a life in MBBS? But really, that is the volume of data you're expected to master. But in 40 years, 2-3 generations have passed. We've really simplified it down for you and reduced the amount of stuff we throw at you in the class. But that doesn't mean the science of medicine has become less. It's actually become more. It only means that more of that has to be dealt with by you. How much time you give, how much effort you give is going to be determined more by you than by us. We will just take out the kernels, the must know, you cannot afford to not know this, those bits, and we will bombard you with that. But we're catering to the base of the pyramid. The middle and the top you have to build yourself. Some of your brilliant students, you'll be finding, I already know this, I studied all this for my pre-med, for my NEET entrance. Give me more, I want to know more. But in my 45 minutes, I do not have the luxury of catering to those brilliant ones among you. So you must have the hunger, the thirst to go and seek it out from textbooks, from reference sites. But it can be very bewildering and it can be very overwhelming. Which is why we did recommended textbooks. Like if you do this much, this one, that is enough to begin with. But there are actually hundreds of titles out there. And some people have written brilliantly and many people have extra points. If you read 3 textbooks of anatomy by 3 different authors, you will get extra points in each one of them. So select it by the way the writing flows, by the style of the author. Select anyone, doesn't matter, and then add the extra points as we tell you in the class. Because my slides have been derived from 6 or 7 books, different resources. So make use of that. Scribble notes. It makes sense to read for it beforehand and then come and write down the extra points, because you've already read it once. You already have a sense of what is supposed to be covered. Okay, so we'll see you tomorrow.